Well, good morning, everyone. Here we are once again. It's Neuro3301. So, welcome to all of those tuning in live, to all of those listening to this the hour before you start the exam. Welcome. And for the 10% or so of people who listen to this to help them fall asleep at night, good night. I hope you have a good sleep and sweet dreams. So this week is our last week of class. We're only going to do two. I believe the university has us scheduled for a class on Friday, but I think that I can say everything I need to say without adding an extra class. So we'll just do the two lectures this week on Monday and Wednesday, and then the customary Thursday office hours. If you have a problem with that, my email address is zach.patterson@carleton.ca, so feel free to send all of your complaints to that address. In the meantime, we'll get focused on the last couple of lectures for class. I'll tell you, I'm just working on the midterm, the final exam now. So I'll have you more details for you now, or I'm sorry, not now, but on Wednesday's class. If you're looking to get started on what you should study, I think that the best way to spend your time would be to review the lectures post midterm 2. But I guess actually maybe, there's going to be cumulative elements to it. So maybe the best thing you could do is review some of the older lectures as well, and look for overarching themes that seem to be present in many of the different classes that I've taught, and consider what kind of questions those could lead to. And that will give you maybe a hint as to where to direct your efforts. So let's get started. How about that snow, by the way? Don't we love just the fact that we have to, it's going to snow and rain, and there's going to be, I got to go out there and snowblow after this. But we love the snow, and probably the end of snow days. I mean, I guess under normal circumstances, we might consider not coming to class on a day like today, but because we're all watching or broadcasting from the comfort of our own homes, we have to keep teaching and we have to keep learning. So that's great. Anyway, so today we're going to talk about the genetics, epigenetics, and how they relate to the topic of sexual differentiation. And this is another one of these topics that it's kind of a grab bag of different items. I don't really have enough, there's kind of too much going on in this field to really give you a overarching survey that takes you from the starting point to the end point and gives you a complete story about what sex is in the brain or what it means for genetics or epigenetics. Instead, I'll give you kind of 3 different little stories that are fairly self-contained, but all work toward the same basic theme, which is that sex, there are sex differences in the brain and in the way the brain wires itself up. This happens under the influence of epigenetics. And like many of the other things we've discovered, there are genetic influences as well, but as far as we know, they're not terribly easy to find. So enough rambling. Let's get into the actual meat of the discussion. So in the early stages of sexual differentiation, you have the 2 different types of gonads, the testes and the ovaries, and they arise from the same tissue embryonically. So the same primordial tissue in the embryo can be converted or can be made to grow into either testes or ovaries, depending mostly on your chromosomal complement. So if you happen to have a Y chromosome present, this will push things in the direction of the testes, and you'll grow these, primordial testicle things, and then everything else happens downstream of that. And if you don't happen to have a Y chromosome, then ovaries will develop as kind of the default condition. The Y chromosome, therefore, is really the key to all of this. The presence or absence of a Y chromosome is what determines whether your primordial gonads will develop into ovaries or testes. And it's kind of an unusual thing because as far as chromosomes go, it's very, very tiny. It's kind of a deformed semi-chromosome. It's very gene poor. There's only about 45 to 73 genes on the Y chromosome in humans, depending on exactly what you define a gene to be. and it also mutates at a much faster rate than other chromosomes, such that if you allow if the current rate of mutation to continue as it stands, the Y chromosome will become unrecognizable in the future just by a high rate of mutation. Now, this allows you to use it for studies of genetics that can allow you to estimate inheritance and family trees and whatnot quite well, because it's very polymorphic. But it's also a place where clearly the selective pressure against mutation is quite limited. And this makes sense in some level. Clearly, the Y chromosome is not essential for life because half the people on Earth don't have one. So whatever these 45 to 73 genes are, you don't really need them to live. There's one particular gene that you need to kind of determine whether you'll develop testes or ovaries. But beyond that, the other genes, they probably aren't really too crucial. Like I say, half the people on Earth don't have a Y chromosome, and they're just fine. And given this, and given sort of the non-essentiality of it, it's actually not surprising really that it mutates at a much faster rate, because mutations happen, but normally they're selected against, because if you have a mutation that disrupts an important part of your genome on an autosome or in the middle of a gene, typically that will cause problems for your ability to survive and reproduce, and thus those genes don't get passed on. But anyway, the Y chromosome is really the secret to all of this. This is what determines whether or not you have testes. And on the Y chromosome, the most important gene is called SRY. This is named because it's the sex-determining region of Y. The protein is the sex-determining region Y protein. And SRY in humans is expressed 6 to 8 weeks into fetal life. And this is around the time when sexual differentiation begins to happen. Prior to this, there's not any real difference between people who have two X chromosomes and people who have an X and a Y chromosome. But once SRY begins to be expressed, this primordial gonad, as shown in this figure, is tipped to either become, if Y is expressed, it becomes the testes, and if not, it becomes the ovaries. And I think it's important to point out that while chromosomal sex is a pretty hard and fast concept, you either have a Y chromosome or you don't, although for those of you playing along at home, you might remember one example I gave you where you don't have a Y chromosome technically, but you can still have Y chromosome genes due to a chromosomal translocation. But for the most part, you either have it or you don't. But sex, in actual in how it's actually manifested, is not binary by any stretch of the imagination. It's not dichotomous that way. The reason you have this sort of discontinuity between the bare fact of chromosomal sex, whether or not you have a Y chromosome, and the very complicated nature of how sex and gender are actually expressed in human life, is because all the sex chromosomes really do is get the ball rolling on sexual differentiation. They don't really have the final say on how sex and gender are expressed. All they do is determine whether the fetus will develop testes or ovaries. Now, and then all of much of the rest of the development happens downstream of that process. So once you have testes, they'll begin to secrete various androgens, testosterone and the like, and those hormones will travel through the body and into the brain and cause all kinds of developmental changes that are consistent with a male type phenotype, and vice versa. If you have ovaries, you'll have female sort of estrogen type hormones. So hormonal communication, as opposed to chromosomal complement, hormones are graded. They come in different levels. They have different timings to them. They have different effects in different tissues. The effects could be big or small. The hormones, there's enough variability there to do a much better job of accounting for how variable sex and gender and all those sorts of things are ultimately expressed. And the vast majority of sexual development takes place under the influence of hormones. And of course, as I said, there's many non-genetic reasons why these could vary. So the brain, which is where we're focused, contains many sexual dimorphisms that will develop under the influence of sex hormones. Like I say, testosterone and estrogen and these kinds of things. This process in humans begins in utero and continues on through puberty. These sexual dimorphisms, really all they are structural and functional features that differ substantially between the sexes. So it would be something like one part of the brain is bigger or smaller in one sex, well, I mean, there's countless of these dimorphisms. We'll focus on some of the most basic ones. The sex hormones can act in an organizational and activational capacity. This is an old kind of dichotomy that is seeming to be a little bit quaint now as a distinction, but I think it still holds quite some good value. Organizational effects of hormones are when a given hormone has, during a critical window early in life, has the ability to shape brain development, resulting in long-term or lifelong changes in how given brain regions are organized, structural variation, sexual dimorphisms, these kinds of things. Later on, after these critical windows of development are over, the hormones act in an activational capacity, meaning that they no longer can influence structural brain development. What they do instead is influence how the brain responds to things. So that, for example, circulating levels of estrogens when you're, sexually mature, past, post-adolescent, those will have, those have to do with things like your estrous cycle if you're a mouse, or your menstrual cycle if you're a human. They'll cause cyclic changes in various aspects of your physiology, but they won't reorganize your brain in a permanent way. Those hormones early in life, during embryonic and fetal development, on the other hand, actually can organize your brain in a permanent way. So the short-term effects are called activational, and the long-term effects are called organizational. And I guess I won't really get into this, but if you think about the lecture I gave you on obesity, I did get into things about critical windows there, in that case for energy balance programming. But the same logic applies to sexual differentiation. There are critical windows for the development of sex differences in the brain, and that means that certain stimuli have to arrive at a certain time in order for certain differences to develop. And I guess what I want to kind of imply, or keep in the back of your mind as I go through all of this, is that because this is such a plastic process that is so dependent on hormones and other kinds of developmental and epigenetic things happening in a certain sequence, there's plenty of room there for variation in how those things are expressed. So if you are trying to kind of understand conceptually the broad range and diversity of human gender and sexual expression, there's plenty of room to explain that using the methods of endocrinology, hormones, and epigenetics and these developmental processes. It's far from being a obvious binary that's determined only by the presence or absence of a Y chromosome, even though the Y chromosome does kind of get the ball rolling on things. There are sexually dimorphic brain regions. There are many of them. Some of them are controversial. They have their proponents and their opponents, depending on just how the methods are used. But there are some that are fairly wide, are quite widely accepted. Sexual dimorphic brain regions would show structural, that is to say anatomical, or neurochemical, that is to say like neurotransmitters might be different, and molecular differences between males and females. So the frontal cortex of the human brain, as well as the hypothalamus and amygdala, are all regions that have a high degree of sexual dimorphism. Within these regions, certain subparts of them are the most dimorphic. One area that's really captivated the attention of researchers is called the medial preoptic area, which has a subregion called the sexually dimorphic nucleus, SDN. So it's sometimes called the SDN-POA. And this part of the brain in rats is about 5 times larger in males than in females. And this is often, or this difference is not due to chromosomes, it's due to the organizational effects of testosterone. In the rat brain, testosterone is converted to estradiol, and that's what actually has the effect, but that's a bit too confusing to get into for our genetics class. The point is that this is a structural difference in the anatomy of the brain between males and females, corresponding to a 5-fold difference in size that develops under the influence of hormones. So testosterone is really to blame for this, and testosterone is produced by the testes. So this is one example of how a structural difference in the brain results from differences in hormones, which in turn result from whichever of the 2 types of gonads you might have happened to develop. And the ball for that is all set in motion by SRY on the Y chromosome. So that's ultimately what we have to kind of ask is, okay, if all of this, if we're trying to answer how does this all start, what gets it going? It's SRY on the Y chromosome. Now, SRY is simply a transcription factor, and it doesn't, on its own, who knows what it really, well, we know what it does, but I mean, like, what's downstream of SRY, I guess would be a better way of introducing this slide. SRY gets the ball rolling, but what does it do? SRY is actually expressed for a fairly short period of time, yet it has so many strong effects. Well, the researchers, so going back a bit, researchers noticed that you could have XY mice that were missing the gene SOX9. SOX9 is located on chromosome 11 in mice and I think chromosome 17 in humans. And if you had an XY mouse, that is to say a mouse that is genetically male, it has a Y chromosome, if the mouse happened to lack SOX9, the mouse would develop into a, anatomically speaking, it would develop into a female. It would still have an XY chromosome, yet that process of SRY turning on and getting the ball started on testicular development, that would not happen. So this is rather an interesting finding because we normally think of sexual development as being something that has to do with the sex chromosomes, the X and Y chromosome. Yet, if you take away a gene on the autosome, chromosome 11, you have this profound effect on sexual development. And similarly, mice with XX mice that have two X chromosomes with abnormal SOX9 expression will develop as males. So it seems as though SOX9 is somehow involved in sexual development at that very early stage of determining which of the two gonads you'll develop. There's a handful of others that are involved in this process, SF1 and FGF9 as well. And ultimately, what researchers found by around 2008 is that these are all part of a network of transcription factors and coactivators and whatnot that work to So SRY and SF1, these two factors cooperatively upregulate SOX9 by binding to a distal enhancer, and I'll explain what that means in the next slide. But what it shows is that SOX9 rather is the gene that's directly downstream of SRY, and it seems to be really the critical factor for actually determining whether or not a mouse will develop as a male. Once SOX9 is upregulated under the influence of SRY, its protein will kind of dimerize or join with SF1. SF1 is another protein that you can have. It's another gene, and it'll ultimately replace SRY at this regulatory enhancer. So SRY comes on for a little blip, a little brief instant of time, but by activating the expression of SOX9, SOX9 basically comes back and sort of, how to explain this? SOX9 kind of binds with its own enhancer after it's expressed and thus maintains its own expression. The point being, anyway, is that you only need SRY to come on for a brief period of time, like a kind of a blip during early development, and once it comes on, it turns on SOX9 and various other things that continue this process of sexual differentiation long after the blip of SRY has passed. So this uses a positive feedback transcriptional mechanism that I don't think we really need to get into in too much detail. But enough said, I guess. There is this very crucial gene located on an autosome that is directly downstream of SRY that really is responsible or crucial, or I guess we should say necessary for the development of the testes. Now, if you need a reminder of what enhancers are, you can check out this slide from the functional genomics lecture way at the very beginning of the course, and I'll put this in here to remind you that if you're looking to study for the final exam, it's a good idea to go and review the earlier material, because these things, it's expected that you'll be able to understand these concepts enough to write about them on the exam, and that might, at this point, you might have to go back to what you heard about in September and remember, sort of remind yourself about it a little. But here we're talking about enhancers, and enhancers were discovered as kind of an interesting counterexample to the earlier dogma that gene expression was regulated by nearby things. So you would have promoter regions of genes. Here's an example of a gene, this blue area. The transcription start site is marked by this arrow, and the promoter region would be marked by this blue space upstream of the TSS. And it was normally thought that, like, gene expression regulation was mostly controlled by things near to the transcription start site, right? That makes sense. I mean, if you're going to turn on a gene, you should start right near where the gene begins to be expressed. But enhancers are distal regulators, and they can exist many tens of kilobases, maybe even hundreds of kilobases away from the transcription start site of the target gene. This would be an example of an enhancer in orange, and enhancers can bind transcription factors, and then by way of the three-dimensional folding of the genome, where DNA that's linearly very far apart can be brought into contact with very far apart areas. Three-dimensionally, and thus influence the expression of those distal areas. So in the case of enhancers, you see the enhancer is brought into contact with the TSS, and that actually enables gene expression. So enhancers are distal elements that regulate gene expression. They physically interact with genes by way of three-dimensional folding of the DNA. So SRY binds to a distal enhancer of SOX9. That's the point here, and that's how SRY is able to regulate SOX9. And I think this is important to emphasize because the undercurrent to this lecture is just how plastic and variable the development of sex differences can be. It's not simply a matter of whether or not you have the X or the Y chromosome. That definitely plays a big part in it, but downstream of the presence or absence of a Y chromosome and the SRY gene in particular, there are so many other things that are really quite flexible, I guess, and epigenetic, and we don't really know for sure what this means in terms of the diversity of human sexual expression, but I think it clearly gives you enough latitude to explain quite a lot of variation just with these mechanisms. So let me show you the power of a single enhancer. We haven't really talked about enhancers a whole lot in this class, but I think it's worth getting into. So here is a map of the SOX9 locus in mice, and this is located on chromosome, what was it, chromosome 11? Yeah, chromosome 11 in mice. Let's see here. So here's the SOX9 gene. This arrow indicates the transcription start site, and they've given locations of all the enhancers they could locate upstream. This scale bar tells you how far up we're going, and you can see we're talking about vast distances. So if we set the transcription start site of SOX9 at 0, you can go up 100 kilobases, and you found several enhancers. 500 kilobases, that's half a 500,000 base pairs, that's half a million. 1,000 is a million base pairs, and the most distal enhancer they were able to find is 1,500 kilobases. So quite a ways upstream of SOX9. Previous understandings of gene regulation would have believed that the only places relevant to regulating SOX9 would be kind of within this little box. But actually, you can go up, millions of base pairs and still find regions where transcription factors binding can influence SOX9 expression. So these are the enhancers. And in 2018, Gonet et al., publishing, I believe, in Science, used DNase-seq and ATAC-seq. Don't worry too much about what these are. All they do is they detect regions of open chromatin, and open chromatin regions kind of characterize enhancers. So to make this very simple, these two methods are sequencing-based methods. They give you data sort of similar to ChIP-seq, but they're not ChIP-seq. They're methods that are basically used to find enhancers. We'll leave it at that. And they found all of these enhancers, and enhancer 13 really caught their eye. This is one that was active in males and in the Sertoli cells of male primordial testes, but not in the granulosa cells, which were seen in females, the sort of female primordial ovaries. So you can see this ATAC-seq peak is rather high in the male Sertoli cells, but hardly any higher than the overall background in the female cells. So this appears to be a male-specific enhancer. and it's located upstream of the SRY gene. The authors also did use ChIP-seq to show that this region is bound by SRY in vivo. I'm not showing that slide, but what we have here is from the authors. It's an enhancer that's located in a region of open chromatin. The open chromatin is only seen in male cells having the SRY gene, and this particular region also binds to SRY. So you have very strong evidence that it's an SRY activated enhancer. And enhancers regulate gene expression. So a very simple test is, what happens if you delete this enhancer? Through the use of CRISPR, which is a gene editing technology, you can come in and basically erase enhancer 3 from the genome. You just take it out and leave everything else in the same place. What happens if you do this? And we're talking about an enhancer that's only about 500 some odd base pairs, 557 base pairs in length. So a microscopically sized enhancer, very, very tiny. The genome is, billions of base pairs. We're talking about 500 base pairs. So a very, very tiny thing was deleted. And what do you get? Well, XY mice that have this enhancer deleted on chromosome 11 don't express SOX9 in the developing gonads. That's all this is showing. It's just a in situ hybridization, I guess, or maybe it's immunohistochemistry. But no, it's an immuno. But anyway, they're showing the SOX9 expression here in green in the developing gonads. You can see in XY males that are either homozygous for the good copy of enhancer 13 or heterozygous will happily express SOX9 in the developing gonads. But mice that are lacking this enhancer and have the XY complement don't express it. So these are mice that are genetically male, whatever that means, and they don't express the correct transcription factors in the developing gonads. And what happens? Well, Lord Almighty, they develop as females. So if you take out this enhancer, which is only 557 base pairs, the mice, despite having a Y chromosome, will develop female. They have all the normal female characteristics. They don't, they're not fertile, I don't think, but they have all the anatomical characteristics. Here's, a wild type male. You can sex mice by looking at the anogenital distance. So this little distance here is what you would use to determine if it's a male or female. If there's a big distance, it's a male. If there's a small distance, it's a female. Females also have nipples and whatnot as well, and various other things. But look, here's a wild type female. That's what she looks like. And here is a XY male lacking this enhancer, and that mouse develops as a female. No researcher who picks that mouse up would really be able to tell the difference without doing some kind of genetic test or trying to breed that mouse or anything along those lines. So really quite an astonishing thing. And humans, it should be said, have a similar kind of enhancer. So we've, you can't obviously delete this thing in humans, but if you look at the, human, genetics, we have a homologous area in our own, in our own DNA, meaning that it's likely that a similar process is playing out in human beings. So I think it just bears sort of emphasis, talking about how flexible and plastic and how dependent on epigenetics sexual development happens to be, to think that all it takes is this 557 base pair enhancer on chromosome 11 in mice. That's all it takes to make the difference as to whether a mouse will go, be developing anatomically as a female or as a male. And I think that's a pretty important point, because if you think about the importance that society places on sex, relative to the very, very, very tiny difference the very, very tiny change you would need to make genetically to completely flip things on its head, it's just a rather interesting proportion to consider. I don't know what I don't know what it means for your own values or your own opinions on the topic, but I just think it's an important piece of data to have in mind when you're pondering it. Okay, so we'll put that aside for now and maybe just spend a moment to review the numerous sexual dimorphisms that exist in the rodent brain. This here is mostly for your own reference, so I wouldn't expect you to memorize these topics in any organized way. Rather, I'd like you to just consider all of these differences and think about their implications for brain development. So there are many things that neuroscientists can measure: the volume of certain brain regions, the density of axons and dendrites, so-called fiber density, the properties of the synapses, whether you're talking about electrophysiology or morphology, branching, that is to say, how arborized are the dendrites, how many branches do they form. More branches is generally taken to be a good thing. If you have nice, thick, bushy dendrites, it means your neurons are making a lot of connections and participating meaningfully in circuits and processing, which is a good thing. Neurochemical phenotype. There can be sex differences in the neurotransmitters used in certain brain regions and the peptides expressed. Cell number is also involved. There are brain regions where sex differences exist in the number of neurons you have or the number of glia, for example. Activational state. How neurons respond to neurotransmitters and their neuroplasticity, long-term potentiation, this kind of thing, as well as rates of neurogenesis can be different. So these are all the things you can that people have measured differences in. And then in the brain regions where they've looked, you've got medial preoptic area, ventral medial nucleus of the hypothalamus, bed nucleus of the stria terminalis, binocular area of the visual cortex, amygdala, lateral septum, medial preoptic, blah, 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 blah. Anyway, hippocampus, frontal cortex, many areas. And rodents, I don't think their neuroanatomy is easier to study, but I don't think there's a huge reason to believe that human brains wouldn't also have similar levels of sexual dimorphism if you were to really look in detail. And this is not to say that male brains and female brains differ in any way that's relevant to our societal values or what we think is important or the moral weight we put on them, only to say that there's plenty of room for there to be variation between people. And these areas do not all have to vary in lockstep. You probably don't just have a female brain or a male brain, like in human beings at least. You probably don't just have a male brain or a female brain. You have a continuum of phenotypes that it sort of biases towards more prototypically male or more prototypically female, and that continuum can be different for every brain region, leading to a very complex behavioral and interpersonal expression. Rodents, I should say, are a good model for understanding the brain and the sex differences there, but it isn't actually clear whether these findings translate reliably to humans. So, you kind of have to take the evidence in hand and weigh the different interpretations. Human brains, on the kind of level of coarse neuroanatomy, actually seem to be rather less sexually dimorphic than rodents. It's difficult to say on a neurochemical level or on sort of the level of microscopic differences that we're able to measure in rodent brains. But if you look at brain scans of humans and the type of anatomy we have access to, sheer differences in size and shape of brain regions don't seem to be as significant in humans as they are in rodents. And this is, I guess, maybe in line with what you'd expect. People really get excited about differences in the brain to say that, oh, the male brain looks like this or the female brain looks like this. In all likelihood, it's much more variable in human beings, not just between the two sexes, but between individuals in general, such that you might have some parts of your brain that are perhaps more one way than the other, although who really knows? I don't think that's ever really been proven. But anyway, all of this is maybe unsurprising if you think about the diversity of human and sex and gender expression and the role of culture in shaping behavior. So I guess maybe a better way of summarizing all of this is to say that for rodents, the sex differences in the brain and the sex differences in behavior are determined to a high degree by their genetics and by their chromosome or by their hormonal exposures. Whereas for humans, a good deal of this depends on culture. If you think about many of the most kind of classically sexually dimorphic styles of human behavior, whether it's clothing or personality or way of speaking, many of these are just culturally conditioned and don't have any biological basis whatsoever. They're just things that people thought of one day and became traditional. So sex differences in the prevalence, but all this being said, there are clearly differences, and there's a lot of room for differences to develop. What's more, there are sex differences in the prevalence of certain mental illnesses. So even if the authors here can't find anatomical dimorphisms, there must be some kind of difference there, because so many mental illnesses are biased toward one sex or the other. And here I've got a list of all of these. I really wish I could go in and study and like kind of describe these in more detail one by one, because I think they're all very worthy of considering. But you can take a look at the slide when I release it and see for yourself all of the evidence that there are for sex differences in the prevalence of mental illnesses and neurological conditions. The citation is McCarthy et al. 2017. Peg McCarthy is a big-time famous researcher in sex differences in the brain, so somebody you should really be familiar with. But you have things like autism spectrum disorder is 4 to 5 times higher in males than females, although there's always controversy as to whether females are simply being misdiagnosed or if cases of female ASD are simply being misallocated, they're being put into a different category of disease. Although far as we know, even in documented cases, you do see differences. I believe I brought this up, but in males, male ASD cases, you tend to see more of the social impairment phenotype, where intellectual impairment is sort of overrepresented among cases of female ASD. Conduct disorder and oppositional defiant disorder is 3 times higher in males than females. Who knows why that would be? That sounds like the kind of thing that could be socially could have social factors involved. Males and females are certainly raised differently in that regard, but I don't really know. I mean, that's beyond my expertise. And ADHD is 2 to 3 times higher in males than females. That's another one where, as kind of an outsider to this exact field, I don't know for sure how this research is done. You would think at least that maybe there is just as much ADHD in females, but it's just diagnosed or it's just missed by teachers, because males, when they have ADHD, tend to act out and become disruptive in class, whereas females maybe don't act out and the teachers don't notice them, and that females are never diagnosed and just have problems their entire life learning that go uncorrected because nobody is there to notice it. So it's one of these questions of like, well, is there really a difference in the actual disease process or the prevalence, or is it just that the symptoms manifest differently depending on whether you're a boy or a girl? All very valuable questions to ask. Some of them I think maybe less so. Schizophrenia is 1.42 times higher in males than females. There, it seems unlikely that there's a secret subset of females who have schizophrenia but don't nobody can see it. That's a little harder for me to imagine, but then again, I'm not a psychologist and I don't have a deep connection to this field. But I think you should at least go through this list and really think about it carefully. For each of these conditions, can you imagine there being a biological difference, or can you imagine a cultural component for it as well? We're here focused on the biology, but biology and epigenetics in particular plays out in the context of culture, so you can never really dissociate the two. So that was kind of a little interstitial to just point to. I guess what I'm trying to point to here is the importance of considering sex differences in the brain and in epigenetics and these kinds of things when you're talking about mental illness, because for many of these conditions, the sex difference is the most salient feature of the whole thing. The fact that certain conditions are much more common in males versus females or females versus males is probably an important clue as to what the etiology of that disease happens to be. Now, let's get back to the mouse brain, because you can tell probably from the way I speak that I'm much more comfortable talking about mice and rats than I am about humans. For one thing, it's sex is a pretty clear concept in mice, whereas it's a much more fluid and complicated topic in human beings. So we can stick with mice and be a lot more certain about what we're saying, and that anyway. So there is this chromosomal complement or chromosomal side to sexual differentiation, basically the presence or absence of the Y chromosome. There's also an epigenetic contribution to sexual differences in the brain, brought about by DNA methylation. So this is another paper from Peg McCarthy's group. This was published in 2015 by Bev Nugent and company. The observation is basically this. Remember the preoptic area, the sexually dimorphic nucleus of the preoptic area? I talked about that a few slides ago. This is an area where if you want to it's a good area to study for sex differences in the brain because it's an area that's very different between males and females. So if you are curious about these, what could be causing sex differences, try to find an area of the brain that's really, really different because it's easier to find these things. So the observation here is that de novo DNA methyltransferase activity is higher in the early postnatal period in the female preoptic area compared to the male. So this plot here is showing the level of DNMT activity, DNA methyltransferase. That's on the y-axis. Higher numbers mean more activity. And then on the x-axis, we have age of the rat in days. So day 0 is the day of birth, and that goes all the way up to 1 week postnatal. Then the lines are colored by the sex and manipulation. So black lines are females, blue lines are males, and then the red line is females who have also been given estradiol for reasons that you would understand if you've taken hormones and behavior class. In the case of rodents, estradiol, which is really an estrogen, actually masculinizes the brain. So you can imagine that it's testosterone, because in real life it sort of would be, but without getting into the endocrinology of it, the red line is a female that's been exposed to masculinizing hormones. And what do you see? Well, the control female has a rather high level of DNMT activity, de novo DNA methyltransferase, and both the male and the masculinized female have much lower levels, at least for the first 2 days of life, from day 0 to day 2. And then by day 4, they're all about equal, and the levels go down with time. So there's this difference in the level of DNA methylation activity in the first couple days of life. And this manifests as higher levels of DNA methylation genome-wide in the female preoptic area, so female POA. Here we have on the y-axis percent methylation going from 0 to 25%. Something's very wrong about this scale, my friends. By the way, if you're looking at methylation overall, it should be much closer to like 75 or 80%, not between 0 and 25. So I don't exactly understand what's happening with this, but we'll assume they're looking at some subset of the genome, maybe CPG islands or something like that. Anyway, the point is that the levels of methylation are much higher in control females, but not males or females that have been masculinized. So the conclusion is that this masculinizing treatment of estradiol brings DNMT activity and methylation levels in line with males. And this ultimately points to an important role, or seems to suggest at least an important role for DNA methylation in the development of these neurobiological sex differences. And note that it's under the influence of hormones, because you can take a rat that is biologically or chromosomally a female and has 2 X chromosomes, and simply by exposing that female to masculinizing hormones, you can change the level of DNA methylation in her brain and make her brain more like the male's brain. All it takes is some hormones, and the DNA methylation will do the rest. So that's what the authors showed. They naturally, when you're in a position like this with this type of finding, you're thinking, well, okay, if females have more de novo DNA methyltransferase activity, and they also have higher levels of DNA methylation in this part of the brain, what happens if we inhibit DNA methylation? What if we block de novo DNA methylation? Will this make the female rats more like males? We already know that we can give the female rat hormones to masculinize her. That's all understood. But that's maybe not surprising, because hormones are precisely what masculinizes males as well. But can you go in and manipulate the DNA methylation levels and achieve the same result? So that's what the authors tried. They gave inhibitors of de novo DNA methyltransferase. The inhibitor is called zebularine, and there's another one they use called RG108. These are they're actually chemotherapeutic drugs that basically block the DNA methyltransferase enzymes. So they're actually pretty nasty drugs. You wouldn't ever want to take them, but you can give them to a rat into the brain by an ICV injection, intracerebroventricular, and block de novo DNA methyltransferase. And what you will see when you do this is a change in the density of dendrites, which is one of the main markers of sexual dimorphism in this part of the brain. So male mice exposed to vehicle have a certain number of dendrites. Females, naturally have lower numbers of dendrites, or I guess dendritic spines to be precise, not dendrites, dendritic spines. But if you expose females to either of these two DNA methyltransferase inhibitors, the number of dendritic spines they have in this part of the brain becomes equivalent to males. They have a male-like level. So you've reversed this sexual dimorphism and made the female rats, I should say, much more like males. This is funny. I used to make fun of Alfie when he was my supervisor for always mixing up mice and rats. I thought, how could you possibly mix those things up? But now in my old age, I find myself doing it as well. So I think it's just a matter of how much experience and how much time you spend having your brain warped by neuroscience that you eventually stop recognizing the distinction between these two types of animals. And worse still, I suppose, is when you lose your ability to distinguish between rodents and humans, which I think is definitely a disease that afflicts many highly advanced neuroscientists. Here, by the way, is just pictures of the dendrite or the dendritic spines. These lines here are dendrites, and these little dots are dendritic spines revealed by the Golgi-Cox method of staining. This method is the kind that gives you those really nice pictures of pyramidal cells in the cortex with the nice shape of the axons and dendrites and whatnot. So you can count dendritic spines with this approach, and they find that if you inhibit DNA methyltransferase, you get a masculinization of spine level in the females. You can also look at the behavior of females that were exposed to this. Recall, by the way, that these were only given during the earliest stage of postnatal development. This is not something that the rats are being exposed to continuously. It's only during the first couple of days of life. And despite the fact that this is a very short-term change early in life, you have these long-term differences in neuroanatomy or histology in terms of dendritic spines, but you also have differences in behavior. So one of the behaviors you can use that would be described as a male type or a male typical sexual behavior is mounting. Mounting is a type of, well, you probably all know what mounting is, but it's just when the male mouse will kind of hop on top of the female mouse or rat or whatever, female rodent, and begin the process of copulation. Okay, so this is a male thing. Female rats don't tend to mount male rats, although it is possible as a dominance display and that kind of thing. But typically, maybe you've seen dogs do this too, but it's kind of funny to watch actually. But anyway, it's typically a thing that a male would do, but not universally. So you can just count the number of mounts in a given period of time, and the number reflects the level of male type sexual differentiation. And you can see that control males engage in about twice as many mounts per unit of time than females. So females are still doing the mounting, but they're just doing way less of it. And if you treat females with zebularine, which is this DNA methyltransferase inhibitor, the number of mounts they exhibit approaches the level of a control male. And in fact, if you treat a male with zebularine, the number of mounts he uses is even higher. So it like hypermasculinizes them, I guess. So all this is to say is that there are structural, morphological differences between the male and female brain, as well as behavioral differences between the male and female brain. And if you're to believe this paper, then those differences are mediated by DNA methylation. And in particular, DNA methylation that's being set up very, very, very early in life, such that if you disrupt that DNA methylation, these differences no longer exist, and the male and female rats become much more similar, and those sex differences disappear. So this is only talking about rats. It's only talking about a very specific region in the rat brain. But if you want to use your imagination, it'll at least tell you that some element of sexual difference or sex differences and sexual dimorphism depends on these kinds of epigenetic processes that play out under the influence of hormones. And as I've been saying, that gives you plenty of room to imagine what could be the source of these persistent differences in sex and gender expression in human beings. The human story, of course, we have no clue what's really happening, or we have very little idea what's really happening. But by doing this kind of work in rodents, at least you lay out some parameters of what you might expect. So if such a thing is possible in nature, then it could be happening in humans. And if it were, it could explain a whole bunch of things. But you'll have to wait and see in the coming years whether it really is happening or not. So McCarthy has kind of taken her life's work and summarized it as follows, which I've only given you because it shows in a more practical way how epigenetics connects with these other anatomical differences. It's possible that you as neuroscientists already kind of understand everything all up here on this chart, this is showing how the sex differences are developed, you probably understand at some level what dendrites are and how microglia can prune dendrites, and how astrocytes play a role in maintaining synapses and all this kind of stuff. And so if you think about on this level, you've got all these cellular and anatomical differences that exist between males and females they kind of play out in the classic neuroscience way that you probably know and love. If you go back one step, you may also be familiar with some of these neuroimmune interactions between, say, prostaglandins and cytokines and histamines and things like that. McCarthy argues that these inflammatory mechanisms actually play a pretty sizable role in setting up the sex differences. The prostaglandins and cytokines especially seem to mediate this sort of neuroimmune based different pruning of dendrites. Microglia and astrocytes can both work to prune dendrites, and so they might be involved in all of this process. And the way these genes are turned on, well, these genes have to be turned on by something. So all of these inflammatory genes are turned on. They go in to activate the astrocytes and dendrocytes, or not astrocytes, and they activate the astrocytes and microglia. And what turns these genes on? Well, they're released from repression, transcriptional repression to be precise. Something is, normally, something in the female brain, these genes are not expressed because they're repressed. And what are they repressed by? What can repress gene expression? Well, none other than DNA methylation. So the argument is that high levels of DNA methylation in the female brain repress all of these inflammatory things and thus change the anatomical development of the female brain. For males, there is less DNA methylation in this part of the brain. This means that the methylation levels at these immune genes are lower. These immune genes can thus turn on, activating the microglia and the astrocytes and causing all of these anatomical differences in the brain to develop. So really the linchpin or the secret here is this epigenetic difference of DNA methylation. And DNA methylation is downstream of hormones. Aromatization is simply the conversion of testosterone to estradiol. This is what happens in the rodent brain to bring about all of this. But anyway, the point is that hormones control DNMT levels, the DNA methyltransferase levels. This controls the DNA methylation. DNA methylation controls the expression of these immune genes. These immune genes set into motion inflammatory processes that activate astrocytes and microglia, bringing about all of these sex differences in anatomy. Does that make sense? It's kind of, she's trying to integrate a whole lot of different things here, and many of these are known, these are known to varying levels of certainty, and of course they only apply, as far as we know, to the rodent's brain. But knowing that a mechanism exists in nature at least tells you that that thing is possible, and it may be involved in other aspects if it is. If it's not, then something else is going on in the human brain. We don't really know. This kind of information would be very difficult to obtain. Okay, so one more topic to get into, and we're kind of going all over the place here because we're trying to cover a lot of different things at once, but hopefully I'm communicating to you that the topic at least has a lot going on. One more question is this: can genetics or epigenetics help us to understand sexual orientation? Because in addition to the bare sexual dimorphisms that exist in the brain and behavior, there's also things that, there's also differences, or there's also variation, I should say, between individuals in the expression of those things. Human sexual orientation would be the biggest and longest studied one. It's very complicated and difficult to define, much less study. What I mean by difficult to define is this: what makes somebody gay? Like, what would the cutoff be if you were doing a study and you had to say, okay, we're going to put all of the heterosexual people here, and we're going to put all the homosexual people here, I'll put all the straight people here, put all the gay people on this side, and we'll take them and put them into two separate groups, and we'll study the difference between them. Do a GWAS study or something along those lines. Where you draw that cutoff line is very relevant to what you're going to be able to find. It's relevant for a number of reasons. One would be that if, sexual orientation is a continuum, very, it's not super common for somebody to be 1000% one side of the spectrum or the other. Usually people are somewhere, it's sort of a bipolar distribution. So when it comes to sort of same versus opposite sex attraction, most people are towards one end or the other. But there's a whole middle ground there where people can have varying degrees of allegiance depending on what their mood is that day or anything else. So you have to kind of draw a line of like where do you define the cutoff, and of course where you draw that line is quite relevant. Where you draw that line is also very dependent on the culture at the time, because it might be the case that in, say, the 1950s, you might have said, okay, well, if you do even the slightest bit of same-sex conduct, or even have the tiniest thought of it one time in your life, you're gay, or you're permanently not heterosexual. Whereas maybe now you might think, well, hey, there's a lot of variation. Everyone is curious now and again. Maybe you have to do it 20% of the time or 30% of the time. Like the cutoff lines can vary, and the definitions can vary, and what we consider to be normal and abnormal varies. And all of this makes it very, very difficult to study. And of course, animal models are not helpful here because animals don't really have this variety of sexual orientation. I mean, animals can engage in same-sex behavior, but we don't know if it's the same as what we see in humans because we don't know if animals can really fall in love in the same way that humans can. And of course, romantic connection is a big part of all of this. So I'm by no means an expert in the complexity here, but it's very clear that this is not a simple thing to look at. It's not just a number you can measure the way you can with height or BMI. You have a much more complicated definition of it. But people have been trying for many, many years, and there is some level of consistency to it. Twin studies, for example, give heritability estimates ranging from 31 to 74% for same-sex attraction in males, and 27 to 76% in females. In both cases, the role for shared environmental influences seems to be very low, but the role for unique environmental influences is very, very high. So here's a study from Langstrom et al. In 2008 in this table here, and they're asking a couple of different questions. Number one, have you had any lifetime same-sex partner? And you can see the rate of concordance here between in monozygotic twins and dizygotic twins. And in this particular question, at least, the concordance is higher in females than it is in males. Then they could also ask you, what is the total number of same-sex partners you've had? Because have you had any? That's a yes or no question. What's the total number? That's a sort of a continuous numerical question. You could say 0, 1, 2, 3, 4, 5, 6, 7, 8, this kind of thing. And there, the actual concordance between twins seems to be rather lower. The shared environment in the case of male same-sex partnerships is rather low. Here they estimate it to be 0% on average. The contribution is a bit higher in females. The unique environment takes up a much bigger amount, over 60% in both cases, at least by this study's estimates. There's variability depending on who you ask. So there's some environmental contribution to this, they would argue, but we don't know what it is that makes these environmental contributions work. But this would at least tell you that there is some level of genetic causality to same-sex behavior or sexual orientation. Now, people have looked at this for quite a while, but it's an area of a great deal of curiosity, although of course it's one that's quite contingent on the societal norms at the time. But Hammer et al. Back in 1993 published one of the kind of seminal studies in this. This is a linkage study of 114 families containing homosexual or same-sex attracted men. And they were able to identify a region on the long arm of chromosome X as being involved in male homosexuality. It's a subtelomeric region. The telomeres, if you recall, are the little end caps on the end of each chromosome. They kind of are those things that shrink with age and seem to be involved in the process of aging and potentially cancer. And so parts of the genome that are near telomeres, near the very end of the chromosome, are called subtelomeric. So they found some level of linkage. I think this is a rather interesting figure. I put this up here because it's from their paper. And so you should take a look at this on your own time and just try to breathe it in, because this is 114 families is not a huge number of families, but it's a huge number. It's a huge amount of work for a linkage study. These are some of the pedigrees here that they show, the black boxes being male same-sex attracted people. But anyway, so they're showing the average age or the cumulative age for the first same-sex attraction that's given by these triangular points. So an age is given along the y or the x-axis rather. So some people report their first same-sex attraction as early as 5 years old. Maybe 20% of people report their first same-sex sort of inklings being as young as 5. But by the time you get to kind of maybe 16 years old, everyone has had some type of attraction to the same sex. The square represents the age of self-acknowledgement. Typically, it takes you a little while, I guess, between when you first notice yourself, notice that you're attracted to members of the same sex, and maybe you realize that's not the most common phenotype, so there's a bit of a delay there between noticing it and acknowledging to yourself that you might be different. And then the age of acknowledgement to others is when you essentially come out of the closet and tell everyone else that you are gay. And this is one number. These numbers, you'd think, these square and diamond shaped boxes, these would be quite influenced by culture, right? Because I think in 1993 these people would have been coming out in the 80s, and that's a very different time to be to be in the LGBT community compared to 2020, let's say. So there's a difference there. But anyway, I just think that's neat to look at. And then the age of puberty is given by this histogram here. So most people hit puberty midway between around 12 or 13 years of age. You can see that same-sex attraction for most people was manifested earlier than puberty. I mean, almost for everybody, which kind of hints to the fact that this is something that's developed before you ever hit puberty. It's something that happens earlier in your life. And then age of the participants when they were actually studied is given by this histogram. So I just think that's a rather neat thing. It's neat to look at these diagrams and think about the life stories that are reflected by these little points on a graph. That the person who experiences same-sex attraction when they're 7 or 8 years old doesn't come out to everybody else until they're in their 20s. That's a long lonely decade they would be living on their own. Probably a confusing one too, because you can imagine what it would be like to have any type of difference in your orientation, say, or gender expression when you're in the 90s or the 80s or the 70s or before. How do what websites do you go to? Like, who do you talk to online to discuss this? You wouldn't have, how would you find anybody to communicate with on the matter? Anyway, so that's, there's some argument there that it's genetic because they found this region on the X chromosome, and you can look at these pedigrees and see that, like, clearly here there's something going on. You've got this one family where you've got two families within this pedigree where both of the male children are same-sex attracted, and you can see that clearly the rate of same-sex attraction is running in this family. In the general population, it's much less common than what you see in these particular families. So clearly it's running in the family to some level, although it's not showing the pattern of autosomal dominance or anything that straightforward. It's just clearly in the family somewhere. Now, this finding by Hammer and company was seminal in the field, and I'm not trying to make a pun with that word, but it had mixed success upon replication. So this region, this long arm of chromosome X, this subtelomeric region, seems to be real. People have replicated it in subsequent linkage studies, but nobody's been able to do the fine mapping and figure out where in that actual region is the relevant gene, or what gene is actually being affected. And of course, we know linkage studies are very imprecise in general and rather outmoded. So people kind of just gave up on the thing and said, well, this area either exists or it doesn't. We can't find it. So they kind of just, as a field, I think, just threw their hands up and say, we give up. Let's move on to a different technique. And so now we're in the genomics era, and we can look at GWAS studies. So the largest of the type came out in 2019 by Gana et al. This was published in Science back in 2019. This used data from 23andMe, as many of the GWASs we've looked at have. It's a very large-scale GWAS study of human same-sex behavior. And here they were using over almost 500,000 individuals. And they measured a whole bunch of different characteristics, as you can when you have these kind of 23andMe type things where people are paying money to sign up for this service and thus are very motivated to give detailed phenotypic data. If you go to, if you get called in to do like one of those SOMA studies, those things that Carleton would make you do to get points in psychology. Like if you're called in to do a study for extra credit in your psychology class, your motivation level to really give up a lot of your personal details is going to be kind of low. But if you're paying $100 to get a genetic test and you know that the accuracy of the test depends on the data you give it, you're going to sit there and do the best possible test you can. You're going to write all the answers down in exquisite detail. So there's reason to trust, I think, the phenotyping data from 23andMe, and they have a giant sample size to boot. So it's probably an okay way of doing this kind of study, although still less rigorous than like an academic kind of control study where you can really bring people in. But it depends on the complexity of the question you're asking. It's very easy to do a study of a simple phenotype where you don't have to do a lot of questions, but when you get more complicated, it's harder. Anyway, so this is the study, and what did they find? Well, with almost 500,000 participants, they found a total of 5 autosomal loci that were significantly associated with same-sex behavior on a genome-wide level. So all know the way these plots work by now. This line indicates genome-wide significance. These dots are all the SNPs, and the height of the dot indicates the p-value of its association. So how significantly is it associated with same-sex behavior? And they only found 5. Some of them were specific to female same-sex attraction. Others were unique to male same-sex attraction. Those are the upside-down triangles. And then 2 of them are only work when you combine both male and female same-sex attraction. They didn't find any loci on the X chromosome, so they're not even showing it here. Normally, when you do GWAS studies, you focus on the autosomes because of the issue of dosage compensation and X inactivation and whatnot makes the sex chromosomes harder to study. But here, they didn't find any genes on the X chromosome where they looked. So that whole business with the XQ28 or whatever it was, XQ28 from Hammer, they just weren't able to replicate that at all. So they're only looking on autosomes. But for the trouble that they did to get almost 500,000 people, they only found 5 SNPs. That's not very many SNPs, folks. That's a very small number of SNPs to explain something that we believe has a fairly high genetic, it has a fairly strong genetic influence. Heritability of same-sex behavior is comparatively low, as we said. Like the genetic influence is around, previous authors have estimated as being between 31 to 74%. So I guess I shouldn't say we know for sure it's highly genetic. 30% is actually not bad as far as like heritability goes in the big picture. If something is 30% heritable, like for example, if you had a type of, brain tumor that was 30% heritable, you'd be very worried about your family history because 30% is quite high for that kind of thing. For something that doesn't really, hurt you in any kind of way, a 30% heritability is not a big deal, but for something that's really dangerous, you would consider it to be quite high. So low versus high heritability is somewhat subjective. But anyway, as far as other traits are considered, heritability of same-sex behavior is comparatively low. So here is a plot showing family-based heritability estimates. These are given by pedigree studies and twin studies. That's measured as H squared. And then SNP-based heritability is measured by GWAS studies. No need to worry about how this is all measured. We talked about narrow sense heritability. That's what's being measured on the y-axis here. And SNP-based heritability is an estimate of narrow sense heritability given by data from GWAS studies and allele frequency and linkage and whatnot. But we don't need to get into how that's all done. But anyway, a value of 1 would mean that condition is 100% genetic. It's completely heritable. If you have the gene, you will guaranteed to get that condition. Anything less means that a lower proportion of its causality comes down to genetics. And you can see that the estimates given by the GWAS methods tend to be relatively correlated with the estimates given by family methods. But same-sex behavior is between 0.1 and maybe, by these recent numbers, maybe 0.1 and 0.3% heritable, although quite variable. So a fairly large component of sexual orientation then, both from the twin studies and from this GWAS study, a fairly large component seems to be environmental, not genetic. We can also look at genetic correlations. Now I think you're used to seeing these. Here we've got genetic correlation on the x-axis. A 0 means there's no correlation. A positive value means the two things are positively correlated, and a negative means the two things are negatively correlated. So the statistical significance is given by the number above the point. So a number less than 0.05 would be considered significant. Some of these are not. Some of these are. And then the color of the point indicates the sex, with like a kind of a turquoise color being male and sort of a bluish color being female. I guess they didn't want to go for stereotypical blue versus pink. Anyway, so you can look at what they categorize as risky behaviors: alcohol use and smoking and cannabis use. I don't know if you'd really call cannabis use risky. That's maybe up to, that's kind of a subjective decision, I guess, but I wouldn't say it's a high risk activity. But anyway, cannabis use, the genetics of cannabis use, are significantly positively associated with the genetics of same-sex attraction. Psychological, mental health aspects, there's not a huge relationship there. Most of these aren't significant. There is a slight positive correlation with schizophrenia, although it's not a very strong one by the standards of this chart. There's also one with bipolar disorder, but only in females. So in females, the genes for bipolar disorder seem to be correlated positively with same-sex attraction. I don't know what that would mean, nor do I know what it would mean to say that ADHD is positively correlated, although it is, as is major depression. Various elements of personality, openness to experience being one. I think that, if you're living in a heterosexual's world, being open to experience is a key element of exploring your sexual identity, I suppose. And so having some tendency toward that would mean that you might be more likely to self-identify as same-sex attracted. Who really knows? Anyway, so a lot of these things aren't really correlated. And I guess you probably don't want to overinterpret these, but it's interesting to think about. There's a very strong positive correlation with number of sex partners, which I might almost kind of think of being more in the risky behavior category than the number or the reproductive trait category. But again, what you call risk depends a lot on your own personal assessment of things. You can have a huge number of sexual partners at very low risk if you take appropriate precautions, but if you don't, then it can be quite risky. So it really depends. But the same would be true of cannabis. You can smoke a huge amount of cannabis and have it be fairly low risk as long as you're not at risk of developing some kind of mental illness and you still go to work and whatnot. So who knows? Anyway, and you also see a positive correlation with the ratio of the second digit to the fourth digit. This is sort of a favorite. This is like the finger length ratio. I think it's the ratio of your index finger to your ring finger. That the length of those two things, the ratio of the lengths of those two things has to somehow is correlated with your exposure to testosterone in utero. So you do see a genetic correlation there as well. Although it's very difficult when you look at this kind of stuff. And the authors are the first to point this out. All these correlations could be spurious on the basis of similar social factors and stereotyping. So whenever you're thinking that this might indicate some kind of biological property, it's every bit as likely that there's a social element to this instead. So can genetics predict sexuality? Well, we have polygenic risk scores that can take GWAS data and estimate your likelihood of developing, say, schizophrenia or cancer or something along those lines. And you wouldn't exactly call it a risk score here because you wouldn't, being same-sex attracted is not a risk. You're not worried about it. It's just a thing that you could predict potentially. So you could conduct concoct a polygenic score, and this could estimate your likelihood of being same-sex attracted. And the authors did go ahead and do this, and they were able to construct these polygenic scores. So in principle, they could use your genetics to predict your likelihood of being same-sex attracted. But the proportion of variability between people they were able to explain was absolutely microscopic. That number is given by the incremental delta R squared. So the model they built could explain 0.2% of the inter-individual variation in whether or not you ever had a same-sex attraction. That's like just an absolutely microscopic amount. So mathematically, I guess it's significant, but in terms of practical implications, it's not. So at the moment, you can't meaningfully predict somebody's sexual orientation just by looking at their genetics. There is clearly more going on there. And so you might ask, well, what about epigenetics? Well, if you think back to our rodent studies, we know there's a role for epigenetics in the development of sex differences in the brain. So that's certainly involved, but we don't have data on that in humans, and it'd be very difficult to imagine what you would have to do to get that kind of data. You would have to wait for people to die and scoop out their brains and look at methylation. And even there, you may not know what's really causal. So people have been excited about the idea of epigenetics for a while. And when I was looking, trying to do research for this class, I found a paper from Eric Villain's. Well, I found an article in Science talking about, we've got a study in twin brothers showing a link between DNA methylation and sexual orientation. So there was promise that this result was going to be revealed to us at the American Society of Human Genetics. showing us an epigenetic basis for homosexuality or same-sex attraction. But when the authors came out with this abstract, they were roundly criticized for being statistically underpowered. So the results were never published, and I had to kind of do some forensic work to figure out what even happened here. But ultimately, they were just highly criticized for their work, and it was viewed to be bad science. So while it was the front page of the, one of the top science journals in the world, it never actually got published and peer-reviewed because it was just too low quality. So there's really no epigenetic studies worth discussing. There's really just nothing to talk about in the world of epigenetics and sexual orientation that would help you improve your understanding of the topic. Right now, as it stands, we have reason to believe that the environment and the things you're exposed to in life play a fairly sizable role in the development of your sexual orientation. And we're talking here about the very simple aspect of just same-sex versus opposite-sex attracted. So we're not talking about any of the other even more complicated kinds of sexual orientation you can have. Those are tough to study because there aren't, most of these things or most of these varieties, there just aren't that many people who express them. In order to do a study, you need hundreds of thousands of people. And I don't know if you're going to be able to find hundreds of thousands of people who are demisexuals or any of the other different types of varieties people are kind of talking about. Not to mention that those different varieties haven't been really, analyzed to death by psychiatrists and mental, and scientists yet. So none of them. Anyway, the science just isn't there to even begin to approach those kinds of questions. We're working on a very, very simple level, and even still, we don't have any data on the epigenetics that would be useful. The EWAS approach, the epigenome-wide association study, is just not going to do it for it, mainly because if the epigenetics are playing out anywhere, they would be in the brain, and so you would need studies of the brain, not the rest of the, not the blood or some other peripheral tissue. So we need epigenetic studies of the brain, we need studies of the developing brain in particular, but that's extremely difficult to do because we'd like to get studies from, say, early life, fetal development or early infancy, or when these sex differences in the brain are being established by epigenetics, as we've said, if you think back to the McCarthy data from a few slides ago. So we would need data from early life. And you can get brain tissue from fetuses. You can get brain tissue from various other kinds of tragedies of life that allow you access to the brains of infants and whatnot. However, we don't know if those infants would have grown up to be gay or not. So you can't take the data from somebody at an earlier time point and use it to predict or to estimate how sexual orientation develops, because they have to live all the way until they're mature to even know what their orientation is going to be. So the timing of it's very very tough. I don't really know how you would ever solve this in human beings, at least, because the expression of the behavior doesn't happen until later. So brain tissue when it's developing is it's impossible to categorize. You don't know what the orientation of that fetus would have been. So if you think about that, it's a very, very challenging issue, and we're a long way off from ever solving the puzzle. But you are free to speculate about what the environmental factors might be that make up the other 60% of the inter-individual variation in sexual orientation. So to summarize, sex differences in the brain, behavior, and disease risk are common but relatively understudied. Now CIHR and NIH are really trying to focus on sex as a biological variable to emphasize the importance of looking at everything in both males and females, whether you're talking about rodents or whatever animal model you're using, or humans. But we're still a long way off from understanding it. And rodent studies don't necessarily translate to humans. Rodents have a lot of differences that may or may not be present in humans. We're not sure. There's a wide spectrum of variation in human sexual orientation and gender identity, and the studies we've looked at only barely scratch the surface of this. You can do a GWAS study like we saw of a dichotomous trait like sexual orientation. You could lump people into two categories. Are you same-sex attracted or are you not? And you can get to a decent level of statistical power that way, but it's very hard to imagine getting similar results. And those results, by the way, weren't even really that impressive. 5 SNPs is not a lot. You can't even really do a gene ontology test with 5 SNPs. It's very hard to do anything with it. But anyway, it's hard to imagine achieving similar results with less common traits or less common versions of sex identity or gender identity, or any of the other millions of ways that humans can be different from one another. So there's a lot of differences out there, but we have no real way to approach it that I can think of. Now, The fact that sexual differentiation seems to depend mostly on epigenetic processes, and that's, if you consider the fact that same-sex orientation has a very high environmental component, the evidence I've shown you that epigenetic manipulations in the rodent brain can completely change behavioral, the behavioral expression of sex and various other things. The SOX9 enhancer being another one that's basically an epigenetic topic. So there's a high level of epigenetic involvement in all of this. So that gives us a framework. It says, if epigenetics are important, we understand why it is that people can be different despite having very similar DNA. That's a very good starting point. But what those differences are, what organs they are, what brain regions they are in, what time points in your life they develop, and under the influence of what do they develop, that's all up in the air at the moment. So the suggestion, once again, is that this is a neurodevelopmental phenotype, as are many of the topics I've given you in class. These are things that happen during brain development and are kind of fixed in place by the time the brain is fully finished developing, but we don't really know what those are. So I hope I've given you a sense of the questions that people ask and the types of things you might want to think about. I'm afraid I don't have any more solid answers for you than what I've given you, but like I say, I hope that I've given you a sense of the questions and a way of, I guess, maybe contrasting the huge amount of variety that currently exists in the spectrum of human sex and gender expression, and the very limited amount of biological knowledge we have that goes along with that, despite the overwhelming suggestion that epigenetics in the brain are probably involved, but in what capacity and where we do not yet know. So that's all I have for you today, my friends. In the next class, we'll talk about the exam. I have to invent the exam and make it before I can talk about it. So as always, I am working till the last moment on these things. I'm sure you're familiar with the feeling. And so we'll talk about that. And then in the next class, we'll talk about, I guess, some of the social and cultural implications of all this work. I've tried to give you a sense of how the findings were, like the work we're talking about and the data we present comes in the context of cultural factors that are quite relevant. Genetics is a very powerful tool and it's been used in very bad ways in the past. And so I think it's important to summarize or to cap off the class with a discussion of the important cultural issues and social issues that manifest from all this work. So that's what we'll talk about next class, my friends, and then we'll be done and we can all go about our lives and hopefully move on and be newly enlightened. So we'll see you on Wednesday, my friends. And until then, I hope you have a great week and try to drive safe if you're going anywhere.